So, thank you very much, Yvonne.
My pleasure.
Now, I'll just begin the introduction of the Ig Nobel Prize for everyone, and then we'll move on to you, if
Please,
that's all right.
please. Yeah, of course.
Thank you very much.
You're welcome.
خب سلام و درود. خیلی ممنون که تشریف آوردید امروز.
And before you start, I would like to show the audience to
Yeah.
Yvonne as well. So this is our audience today, Yvonne.
Hello.
That's good, everyone.
Hello. Hi.
Yes, I think you froze for a second, but it's all right.
Yeah, it's the
Yeah.
limits of technology.
Yeah, of
I'm
course.
doing my best, I promise.
Yeah, it's fine. It's okay. So, Ali, take it away.
خب اول مقدمه میدم از جایزه ایگ نوبل که چی بود، چی شد و چرا این اصلا خب ما جایزه ایگ نوبل رو داریم و جایزه ایگ نوبل در واقع همچنین یک ویژگی دیگه که داره این هست که خب این یک بازی با کلمات هست که نوبل میشه شریف و ایگ نوبل میشه عذر میخوام بیشرف و همین این جایزه یک بار شوخ طبیعی خیلی زیادی داره و این در حقیقت شروع این جایزه هست. دیدش این هست که اگر قرار کسی این جایزه را ببره، اول هر تحقیقی میخواد انجام بده، وقتی که افراد این تحقیق را میخونن، نتایجش رو باهاش آشنا میشن، اول باید باعث بشه که بزنن زیر خنده. برای نمونه اینکه روی سس پاستا تحقیق کردن یا اینکه اولین جایزه صلح ایگ نوبل به پدر بمب هیدروژنی اعطا شده پس از مرگش البته. و بعد از اینکه افراد خندیدن باعث بشه که خب حالا فکر بکنه که چه جالبه، چرا این در واقع حائز اهمیت است. پس این شرط اعطای این جایزه هست و در نهایت خب خود جایزه خیلی بزرگی هست. 10 تریلیون دلار ارزش داره. 10 تریلیون دلار زیمبابوه که معادل 40 سنت هست و امسال به خاطر تورم این جایزه رو اعطا نکردن به برندگان و فقط یک عدد دستمال محسوب از این مربعی‌ها که توی هواپیما میدن اعطا کردن به برندگان. و با این مقدمه حالا می‌ریم که صحبت‌های ایوان که یکی از برندگان جایزه ایگ نوبل فیزیک امسال هست را بشنویم. خیلی ممنون.
So, I'd like to check. Do you have my audio right now, Yvonne?
Yes.
Great. And if I want to translate what Ali was talking about, he was explaining the whole, Ig Nobel Prize thing, and the fact that due to inflation, you couldn't have the $1 trillion, $10 trillion.
We were so sad about it.
Yeah, and you were given a issue, but
We didn't even get it. We didn't get it. Only the Nobel Prizes got it.
But they showed it in the conference.
But we didn't get it. They kind of forgot. We really wanted our $10 trillion.
Fabulous. به نظر میاد همون دستمال مرطوب رو هم نگرفتن. So I think we're ready for you to start your presentation, and yes, please take it away.
Okay. So hello everyone. I hope you can hear me loud and clear. First of all, I'm very happy to be here, and I'm really happy about your enthusiasm. So I hope I won't. I will be up to the standards. Okay, let me share the screen. Okay, share. Okay. So can you see my screen?
I think I have to adjust that right now. Let me see.
Maybe not.
I think we don't have your screen at the moment.
Wait.
Yes, it's fine now.
Can you see me and my screen?
Yes, everything is all right.
So, hello again. As you probably already know, like we won an Ig Nobel Prize for a paper on Cacio e Pepe. And as my colleague said on the stage, I know that this work kind of reinforces the stereotypes about Italians that only think about food and that we are not very serious, but like the paper is actually very serious, and there is a lot of hard science and equations and stuff like that. So in order to convince you, I would like to start this presentation by showing you a little bit of good old mathematics and introduce you to the main equation of the work. So Cacio e Pepe is a recipe that basically involves 4 ingredients. The first one is pasta, and please don't underestimate the shape of the pasta, because that's very important for maximizing the adherence between sauce and the pasta itself. Then after the pasta, of course, you need cheese, and in particular pecorino romano. Some people also use a parmigiano, but I mean pecorino is the traditional one. I guess like pecorino, Italian pecorino can be difficult to find in some places of the world. So, I mean, it can be done also with other cheeses. But if possible, please use pecorino romano. Then pepper, of course, because cacio is the cheese. Oh, sorry. Cacio is the cheese. Pepe is the pepper. And then there is another ingredient, which is the pasta water. So the pasta water, as I hope I will convince you throughout this talk, is a underestimated ingredient. Probably not in Italy, but in other places of the world, definitely yes. And it's really the key to make a sauce that remains stable and that remains homogeneous throughout the whole cooking process. So in particular, the pasta water is a starch-enriched water. So starch is a complex carbohydrate that is contained in pasta. And when you cook the pasta, starch is released inside the water. And in particular, it's gelatinized. This gelatinized pasta, the water, gelatinized starch in water is really key to reach a stable sauce, but I will talk more in detail about this later. For the moment, the important thing that you should have in mind is that the whole process can be summarized by trying to create a sauce from cheese and water, so an emulsion like this, that should ideally coat the pasta in order to get something like this. Can you see my pointer, right?
Yes, we can.
Okay, so if I show you this, you can see that this is what you should get. So homogeneous sauce coating the pasta, and this sauce at the end of the day is nothing else than water and melted cheese. So the problem here is that this cheese sauce at a certain point should be put in contact with the pasta. But the pasta has been cooking in boiling water. So the temperature is around 100 degrees. But what happens is that at 65 degrees, the pasta, the cheese sauce, actually undergoes a phase transition from a homogeneous state like this to a separated state like this. And this is well known in the cooking, like from everybody who tried Cacio e Pepe, I think at a certain point becomes sadly familiar with this phenomenon. And so this is the problem. Like, I mean, it's not necessary. This is the physical phenomenon that we wanted to characterize. And of course, then at the end of the day, from a practical point of view, a phenomenon that we want to avoid. But just to be clear, this is what we like to call the Italian's nightmare, when the heat actually is such that instead of getting a smooth sauce like this, you get a stringy sauce like this. So I hope it's clear from the images that this is better than this.
Yes.
And
May I provide a short translation for our audience from
yes,
you know
of
the
course, of course. Yeah,
Yeah.
you go.
خب اولش داشتن از این شروع می‌کردن که کلاً پرسه درست کردن یک پاستا به شکل پاستا خیلی مرتبطه، اینطوری نیست که هر پاستایی صرفاً همین نتیجه رو بگیرید. و بعد وارد این شدن که اصلا چرا این پاستای خاص به خاطر اینکه خیلی محتوای ساده‌ای داره، صرفاً پنیر، آب و خود پاستاست. که حالا داشتن اول توضیح میدادن که مثلا یک پاستای خاصی مثل پکورینو رومانو رو ما امتحان کردیم و رسیپی اصلی هم در واقع همون هست. اتفاقی که میفته اینه که شما آبی که پاستا رو توش قرار دادید، محتویات خیلی مهمی که داخلش داره نشاسته یا استارچ هست. این به شما کمک می‌کنه که یک ایمولسیون داشته باشید، همونطور که اینجا دارید می‌بینید که قبل از رسیدن به دمای 65 درجه همین چیز اوکیه، کاملا همگنه، هوموجنس، همگنه. و ولی وقتی که مثلا شما از کاسه خیلی داغ استفاده می‌کنید و میرسه به دمای 65 دیگه اون حالت ایمولسیون و نرم خودش رو نداره و تبدیل میشه به یک چیز سفت توده‌ای. از اینجا قراره اینکه صحبت کنیم که چی کار کردن که این اتفاق نیفته، دیگه اون گوله شدن نباشه توی خود سس پاستا. Yes, Ivan, you can continue.
Thank you. Yeah, please, in any case, interrupt me whenever you want. Like, as you can see, it is not a very formal presentation, so like it can be also a discussion. It's totally fine. Okay.
Yeah,
it's not
thank you.
Formal too much. Okay, so like this process here of the sauce becoming like separated and transforming from a homogeneous state to a separated state is due to a physical phenomenon which is protein denaturation. So again, like this is not a very serious talk, and this image was not done with AI. So like protein denaturation is what determines this protein aggregation. And in particular, we have two proteins in the cheese, which are whey proteins and casein. And when heat is applied to this system, whey proteins denature, and also like the aggregation between micelles is enhanced. Another important thing is that casein in general is an emulsifier. So for example, milk is an emulsion of water and fat, which does not separate because casein acts as an emulsifier, binding both fat and water to keep the emulsion stable. With heat, casein loses its emulsifying properties and also tends to aggregate more. So like you have these two competing effects, one that you cannot emulsify anymore, and on the other side, proteins that aggregate, which determine indeed this phenomenon of aggregation that we are trying to characterize in this work. So, in particular, as I anticipated to Ali, if I'm correct, in our institute where I work, there is a lot of attention to this branch of physics called phase separation. So, of course, like the beginning of everything was indeed trying to understand this in terms of phase separation. And in the original talk that we gave, so I used some old slides, I apologize for that. We had like a whole section about phase separation, but I don't feel very comfortable in talking about it because I'm not the expert of that. So I will just try to show you some very quick overview of what phase separation is. And just to summarize, it's the realm of physics that tries to explain why oil and water do not mix at normal temperature. So I think we are all familiar with the fact that if we put water and oil together and try to mix, then we will find these tiny droplets that at a certain point coalesce and we will have a separated state. So this is also very important in cells. So there has been a very big breakthrough at the beginning of the 2000s, where people have realized that inside a cell, in addition to the organelles that have a membrane, we also have these membraneless organelles, which are formed thanks to phase separation. And this is important because, I don't know, am I becoming too technical here? Please, guys, stop me if I'm becoming too technical here.
By all means, no, everything's all right.
Okay. So, for example, a cell that is going around and the environment changes, there is an infection. Maybe you immediately need some new components to face the infection, but you don't have the organelle ready for that. I mean, maybe you don't have the right molecules to fight the infection. You can very quickly build a very small organelle by driving the system to a state where some proteins phase separate. And once they phase separate, you have proteins all together that can do stuff and produce, I don't know, weapons against the infection. And this is very quick, so you don't have to build a membrane. And this can be done by tuning, for example, temperature, acidity, or other physical variables to actually trigger the phase separation and do it very quickly. So this is the reason why people are interested in phase separation nowadays. I personally was never particularly enthusiastic about at least the mathematics used for that. But I'm happy to say that from today, we can also try to understand Cacio and Pepe using phase separation. And this is what I'm going to talk about now. So before diving into the real experiment, maybe you want to translate what I just said so we can start with, let's say, more...
I was just trying to propose that. Yeah, sure.
Please, go.
این قسمتی که داشتن در مورد صحبت می‌کردن در مورد فاز جدایی بود، phase separation. و چرا اینکه این مهمه هم توی بحث در مورد پاستا و هم در کد. مثالی هم که زدن مثال روغن و آب بود دیگه. روغن شما هر چقدر توی آب بریزید و هم بزنید یک مخلوط واحد به شما نمیدن. این پروسه خیلی مهمه حتی توی ساختارهای زیستی، مثلا سلول‌ها. ما یک سری سلول‌هایی داریم که قشا دارن، ممبرین دارن و یک سری سلول‌هایی داریم که این قشا رو ندارن توی سیستم بدنی مثلا. اون سلول‌ها اگر که بخوان در برابر حمله یا یک میکروب یا هر چیزی که هست از خودشون دفاع بکنن، باید دچار این فیز سپریشن یا جدایی فاز بشن تا بتونن اون حفاظتی که باید رو از خودشون انجام بدن. این مثلا یک مثال زیستیشه که ما توی خود سلول‌های زیستی می‌بینیم. الان میخوان وارد این مرحله بشن که ژلاتینی شدن نشاسته چه کمکی یا اصلا چرا این ژلاتینی شدن نشاسته به ما کمک میکنه که اون مخلوط پاستا رو بهتر درست کنه. So yes, Ivan, you can continue.
Okay. So before, as a last introductionary slide, I would like to say a few words about starch gelatinization. So starch, again, spoiler alert, is one of the main protagonists of this story. Is a complex carbohydrate, and in particular, there are two main type of starch, which is basically a chain of glucose components. And so these molecules have a lot of nice properties, among which there is this gelatinization transition. So, I don't know if you have ever seen powdered starch. I mean, probably yes. But I mean, some people don't know that you can find it at the supermarket. So, this starch, powdered starch, when you buy it, is in a micellar form. So you have these aggregates of starch that sit there and they are stable. Then you put it in water and you increase the temperature. What happens here is that these micelles start to break because of the heat, and all these molecules start to absorb water. So basically what happens is that these molecules that are all entangled very slowly, or not even slowly, like they just start becoming longer and longer and stiffer thanks to the water absorption, until at a certain point you have like that these long molecules start attaching to each other and form a network. So this is a very interesting transition where from a, let's say, cloudy and normal viscosity water, you get to something that really looks like a gel. And this happens at 65 degrees. Interestingly enough, it's the same temperature at which protein denaturation happens. And this is also what happens in the gel that in the pasta water. So pasta releases starch in the water and it's gelatinized. So in order to study the physics behind Cacio e Pepe, we have to study the effect of starch on the stability of the sauce. But because pasta water is very difficult to control, like how much starch it's inside the pasta water. It depends on the pasta. It depends on how much water you use. We decided to use gelatinized starch starting from powder starch, which we can really control the amount, like just with a scale. And in particular, once you gelatinize the starch, you get something like this. I hope you can see that this water is it's not cloudy, but it's not like, it doesn't look like normal water. And this is very viscous. By the way, this is also a non-Newtonian fluid if you do it properly, because like if you poke it very slowly, then the finger goes in. But if you give it like a big hit, then it acts like a solid. So this is very interesting. But in any case, we use gelatinized starch to test the sauce stability in different conditions, just for the fact that we can control the amount of starch. And this is, as you might imagine, like a important thing from a scientific point of view. Now, in order to study the phenomenon, we also need some experimental apparatus. And it's very funny that all the authors were theoretical physicists. So like we really gave our best to build this experimental apparatus. And so this is the sketch. And in practice, there is a pot here containing water. And this water, the temperature of the water is controlled by this object here, which is called a runner. You can cook all kind of stuff using this thing. It's you use it for it's called sous vide cooking, or like a low temperature cooking. You use some vacuum sealed food and you put it in this bath at a certain temperature. You cook it for hours. It's very interesting. I love cooking with this thing. But in this case, it's just an object that keeps the water in this pot at a fixed temperature. And then you have this small pot, which is in contact with the water, which is actually where you put the sauce. And then we have another thermometer, which gives us the temperature of the sauce at all times. Usually there is like a 5-degree difference between the temperature in the pot and the temperature here. So this is why you need the 2 temperature probes. And then once you reach a certain temperature, and so we know, for example, the starch concentration, because this is what we did, like putting a certain sauce with a certain starch concentration, we reach a certain temperature, and then we want to see what is the state of the sauce. So we take a little bit of sauce from here, and we put it on this petri dish, and we take a photo. And then we analyze the photo, and we will do some considerations, but this is in one moment. So this is what we actually built. It's stylish, I would say, but most importantly, it worked. And in particular, again, boiling water here, we have gelatinized starch, we have the cheese, everything is put here together. Oh, important, the sauce gets blended because we really want something that is super smooth. We don't want impurities, because impurities can trigger the aggregation before it should happen from purely thermodynamical reasons. Then once we have our sauce, we put it in the petri dish and we take a photo. And the photos look.
Sorry, may I ask a question? How do you prevent the impurities that you were talking about?
Again, like you take the sauce and you blend it as well as you can.
Mm-hmm.
And of course, there will always be some small impurities. Also because, again, the lab was my kitchen, so
yes,
the standard. But I think like if you blend enough and then you visually check that there are at least no big chunks, this is already good. The beginning we were not doing it, and like the results were very fluctuating. But once we standardized, this kind of worked. You use an immersion blender or whatever you prefer. So actually here, instead of these, we should have put an immersion blender. But yeah. And then we get these. So these are the samples. So these are an example of a sample with small chunks. You can see that this is more or less a good sauce. This is very small. It's like half a mil, I don't know, 2 millimeters. This is not good. So you see here you have a lot of water around and a very big chunk. So this is like an example of what we can see through this apparatus. Maybe you want to translate a little bit what I just said?
Yes, sure. خب اون قسمتی که داشتن اول در مورد نشاسته میگفتن و ویژگی‌های مشخصی که چرا اینقدر خوبه، مثلا اینکه کربوهیدرات پیچیده است، یک زنجیره طولانی از گلوکوز هاست و ویژگی دلچسبی داره اینه که ژلاتینی میشه وقتی که توی آب قرارش میدید. مثلا حتی مثال زدن که از توی سوپرمارکت هم برید پودر نشاسته بخرید، دقیقاً همین اتفاق میفته و همین پودرها، دونه‌های پودرها در یک حالت مختلف یکسانی قرار داره و شما مثلا اینو اگر که داخل آب داغ بریزی تبدیل به یک ژل میشه. این خیلی خوبه و در پروسه درست کردن پاستا به ما خیلی کمک میکنه و ویژگی خیلی خوبی دیگه هم نشاسته اینکه ویسکوز هست و یک سیال غیر نیوترونی، یعنی شما راحت میتونید انگشتتون رو به آرامی درش شروع بکنید، ولی وقتی که با فشار بهش وارد میکنید مثل اینی که مقاومت میکنه. این از ویژگی‌های نشاسته بود و بعد داشتن آپاراتوس و دستگاهشون توضیح میدادن و یک نکته که اینجا گفتن این بود که اینها یک مشت فیزیکدان تئوری هستن، اکسپریمنتالیست نیستن، فیزیکال تجربی نیستن و داشتن گفتن که با تلاشی که داشتیم کردیم که یه دستگاهی درست بکنیم که کار بکنه و توی اون عکسی که نشون دادن 2 تا آپاراتوس داشتن. یکی برای تصویربرداری بود که اولش حالا در قسمت بالا گوشی قرار داره، صرفا حتی گوشی تلفن آزمایش و مقاله رو نوشته، گوشی قرار داره که ازش عکس میگیرن و سمپل و نمونه هر سس رو میذارن زیر دوربین گوشی. یک آپاراتوس دیگه که باش سس رو درست میکردن چند تا قسمت داشت، من دقیقاً یادم نیست چی بود، ولی از هر آب پنیر و نشاسته اضافه میشد، دما رو اندازه‌گیری میکرد و بهتون میگفت دقیقاً در چه دمایی شما چه نوع مثلا همگنی دارید توی اون سسی که درست کردید. اول در واقع اون سس رو درست میکردن و بعد نمونه‌ها رو زیر اون دستگاهی که دوربین داشته قرار میدادن و بررسی میکردن. So yes, Ivan, you can continue.
Oh, by the way, one question. Like, am I getting interrupted by the connection sometimes? Like, happen here during when I'm
from
talking?
time to time. It's not really regular, but it happens from time to time. But it's all right. We can understand.
If like a big chunk is lost, please let me know. Like again,
yeah, sure.
You would expect Germany to have a great internet. It's full.
Yeah, it's fine. It's all right.
Okay, so from here, everything. So this is the experimental apparatus. These are the results that come out from the experimental apparatus. Also, we make photos with our phone. So like really high technological standards here. But then what if you take these photos, you go in a scale of grays. then you can, how do you say, not sequence, but segment these clumps, as you can see. Like you can really see them. No, how can I say? Yeah, you can segment and individuate them in this way. We were very lucky that one of the authors is very good at image analysis. So this was not possible at all without him. And once you have you found these clumps in this way, what you do basically is to approximate them as ellipses. So here we took the most ellipsoidal looking clump that we could find, but in general this can be done, like to approximate with an ellipsis. And then we had to define a size of a clump, and we decided to take the major axis of the ellipses. So this is not, let's say, so this is arbitrary in the sense that we could have taken the area, for example. But at the end of the day, we thought that, I don't know if you remember one of the first images of the pasta becoming, how can I say, becoming stringy. So the stringy pasta is actually what convinced us that maybe it's the length that should be used to, let's say, quantify the size of these clumps. And once you have the size of all the clumps in a sample, you take the average, and then we have an average size. And this is what we use as an order parameter of this sauce. With an order parameter, what you can do is to build a phase diagram. Hello, my flatmates just woke up. By the way, one of the flatmates is also an author. Yeah, sorry, like Saturday, it's not a working day here, so I have to be at home.
Yeah, very bad. It's fine. It's all right.
Don't worry. I don't consider this work. It's a pleasure.
Yep.
So once we have this order parameter, we can build a phase diagram. So here again, we could control the amount of starch in the sauce. So this is the x-axis, and then we can control temperature. And then, for example, you can see that below a certain amount of starch, which is more or less 1% with respect to the mass of the cheese, at a certain, you see, 65 degrees is the temperature where I said that protein starts to aggregate. And then you go up until you get this big clump, and the sauce is ruined. If you put a little bit of starch, you see this thing happens a little bit later, but it still happens. So in this region, basically you have these gigantic clumps, which we called the mozzarella phase. And funny thing, like we were calling it like that for during our experiments. Yeah, we have the mozzarella now. We have the mozzarella. We are in the mozzarella phase. And then we were writing the paper, and we said, yeah, but do we actually write it in the paper that it's the mozzarella phase? And then we said, yeah, let's do it. I think this is a very good idea. People loved it. So I think this is one of the this is our major scientific achievement, to give a name to this phase and call it mozzarella phase. The idea here is that if you go above this threshold of 0.5 degree, 0.5 percentage of starch with respect to the cheese, you see that you have no mozzarella anymore. What you get is these very small clumps, very small, like smaller clumps, and the sauce is more or less stable until 95 degrees. So this is what should like project you to the recipe, saying that if you use enough starch, the sauce will never clump, or at least if you don't do weird things to the sauce, it should be stable also at high temperatures. Usually when you use pasta water to do the sauce, you are in this region here. This is why you're getting these mozzarella-like clumps. And here it's just a smoothing, just to more or less visualize that as you increase starch, the occurrence of these clumps is more and more delayed. So this was at a fixed cheese-water ratio. but you can also fix a starch ratio. So you fix like the certain amount of starch with respect to the cheese, which is to 1%, and then you change the amount of water. So here you see there is 200, like there is double the amount of water with respect to the cheese, and here there is much less. There is less water with respect to the cheese. And if you study the size of the clumps using these mean aggregate size, which is the order parameter that I told you about before, you really see a non-monotonic shape to this curve. Like we had to fix a boundary here, where here we have blue and here we are red. This is totally arbitrary. It's also a little bit for let's say image purposes, that you have a red above and the blue below. But the important thing is that you have this non-monotonic behavior, which is typical of phase separation systems. And maybe you want to translate this, because like here is getting a little bit technical, and so I don't want to lose anyone.
Yeah, sure. Yeah, sure. But let me check first. شما رو متوجه شدید؟ من یه قسمت‌هایشو می‌خوای تو بگی. این من زیاد چیزی یادداشت نکردم. آره تا یک وقت داریم. خب داشتن نمودارهایی که با استفاده از اون آپاراتوس و دستگاه در واقع به دست آوردن نشون میدادن. در واقع یک نمودار فازی بوده که بر حسب دو تا پارامتر بوده. یکی این دماست و این یکی درصد نشاسته است. و چیزی که دیدن مثلا یک همچین حالتیه. این قسمت رو که دوباره به شوخ طبعی گفتن این قسمت میشه مزاردا فیز. باز مزاردا که این اسم خودشون درست کردن و در نهایت هم در جامعه علمی این اسم پذیرفته شده و گفتن که این یکی از بزرگترین دستاوردهای این مقاله بود. خلاصه که در این قسمت اون فازی که ما نمیخوایم وجود داره. هر چقدر که درصد نشاسته بره بالا و همچنین دما هم بره بالاتر، می‌بینید که این قسمت‌های آبی وجود داشت توی این، یعنی که اون فاز گوله شدن هی داره به تعبیری باید تعبیر میفته. هر چقدر که این ادامه پیدا بکنه، هر چقدر که این درصد نشاسته بره بالاتر، این اتفاق کمتر میفته و مثلا اینجا دمای 65 درجه است که شما میتونید برید کامل این چسبیده شده. و همین. حالا ما یکم باید زودتر ادامه بدیم چون که گوگل میت یکم محدودیت داره، تا یک ساعت بیشتر نمیتونیم داشته باشیم. ولی yes, Yvonne, okay. Yes, Yvonne, I translated this part, but I think we only have kind of 10 minutes left on our so yeah, if you could just wrap it up.
I think we can do it. Okay,
yeah,
so
great.
Now we would like to describe this in terms of a physical model. An important thing is that the system has a lot of components. So we have the proteins, we have the salt, we have the water, calcium, fat. So like this is very complicated. So let's say that in order to simplify a little bit, we say that one component is the proteins, whey and casein, because they determine the phase separation, and then we have everything else. Which means that we can describe this as what is can be considered as the binary, like the harmonic oscillator of phase separation, which is this guy here. So it's a binary mixture. And probably, yeah, because we don't have that much time, but be aware that there is a free energy, and the free energy determines a region where pressure and chemical potential are in balance, which is exactly a region where the phase transition occurs. So this is why we wanted to fit the last experiment that you saw to the theory, just because we were theoreticians and at least a little bit of theory should have been done. But yeah, maybe I leave a little bit of technical details here, like just know that we did this and it kind of worked. But I think there is a more interesting part to discuss, which is there is also this part here. But if we have 10 minutes, maybe. Okay, just to show you that there is another object that prevents phase separation, which is this 3-sodium citrate, and it works very well. It's a salt that basically prevents the casein to clump together. And as you can see here, basically, if you use like 2 to 3% of trisodium citrate in your mixture, you basically don't get separation ever. Like it's incredibly stable, and this is what people really use in the industry to prevent cheese and protein aggregation. The only problem is that, at least to me, the taste profile of the cheese is really changed from this salt, and to me it tastes like this American cheese that I really don't like. So I wouldn't suggest to use this salt to stabilize the sauce, because it really changes the flavor. I would stick to the gelatinized starch, which is pretty good at stabilizing and basically doesn't change the flavor at all. So just to wrap it up, because we are late here, let the practical take home message is that if you wanna do a pasta and avoid the dreaded mozzarella phase, you should aim to have this amount of starch in your sauce. And in order to do so, instead of using pasta water, just use gelatinized starch, and which has to be precisely measured and gelatinized. Once you do this, you take the gelatinized starch with the cheese, you blend it together in such a way that you get a very, very smooth sauce. And then you mix it with the pasta by being at least a little bit careful to not put it in contact with pasta that has just been drained. So you take the pasta, you drain it, you wait 30 seconds, you can put it on the stove, but for the first time I would suggest don't put it on the stove, and just mix everything together, and the pasta will be very good. Guys, I cannot stress how many people asked me to do this pasta after we published the paper. Like I cooked more than 20 kilos of pasta, like a lot. So
yeah, we actually read that in the acknowledgement part of your paper, and that was really funny for us.
Like we cooked so much pasta after that. And one of the authors' cholesterol was very bad at a certain point. Like, this is another story. But yeah, in any case, very important thing, the recipe really shines at large batches. This was the starting point. Like, how can you cook cacio e pepe for a lot of people, where you have a lot of heat to handle? And this one big batch that we did, this was the very first big batch that we did. And as you can see, there is no clumps. It just worked. This is a little bit of authors. This guy here is the guy whose cholesterol was not very, very good at the end of the experiments. This is our Roman guy, because like this is a Roman pasta dish, so it's important that we had a guy from Rome. This is the guy that did the image analysis, and then yeah, like people really liked it. They decided to report it on some big newspapers, along with some less important issues going on in the world. And this is the grand finale. You probably saw something, but not everything. Particular, this is the photo of all the people that came with us to Boston. This is my mom, this is my dad, there is also a grandma here. And so when we said, I don't know if you have seen it, like ciao mama, is because our moms were actually there. And so this was the group. And I would like to thank you for your attention, for your enthusiasm. And I hope that one day you will try our recipe. And please let us know, because we are still collecting data of people that actually tried the recipe. And yeah, thank you.
Sure, please, thank you. Yes. Yeah, I'm just going to do a short translation right
Yeah,
now,
please
and
go.
then I will continue again. So, یک قسمت من یادم رفت، اون قسمت ایمیج پروسسینگ و اصلا فرآیند تصویرشون بود که خیلی فرآیند قویه. در واقع این از چند تا کد استفاده کردن برای اینکه یعنی در واقع کار خیلی شایعی هست، ولی اولین باری که این کار کردن اینها که دقیقاً اون کلامپ ها و اون گوله ها رو پیدا بکنن از اون تصویری که گرفتن. شعاع بیزی مثلا اگر که شکل بیزی داره شعاعش رو پیدا بکنن، اندازه‌ش رو بگیرن و همینا. یک قسمت پردازش تصویر داشته که حالا توی عکسشون نشون دادن. Ivan, could you show us the picture you had from your yes, this one exactly. Yeah. مثلا این آقا قسمت پردازش تصویر رو انجام داده. اون کسی بوده که در واقع کلسترولش خیلی بالا بوده به خاطر اینکه از دست پاستا امتحان کرده. و کلا استارتینگ پوینت و اون قسمتی اصلا شروع شده که اینا به فکر این پروژه بیفتن این بوده که به مقدار زیادی این پاستا رو درست بکنن. یعنی یک مشت دانشجوی خوابگاهی بودن که مثل هر دانشجوی خوابگاهی دیگه سعی می‌کردن یک پاستا درست کنن، یک چیز درست کنن تمام شه بره و بخوان به کارشون برسن. این پاستا خب خیلی ساده است. محتویاتش فقط پنیر و پاستا و آب همونطور که گفتم. و فلفل که اینها رو در حجم زیاد انجام دادن و درست کردن یک سس خوب عملاً غیر ممکن بوده. این قسمتی بوده که اصلا باعث شده که اینا به این پروژه فکر بکنن و مثلا مقدارش هم نوشته بودن که شما 5 گرم نشاسته نیاز دارید. مقدار توی خود مقاله هم هست. اون رسیپی رو حتی میتونید توی مقاله ببینید. 5 گرم نشاسته نیاز دارید. 2.5% میزان آبی که دارید استفاده می‌کنید پنیر و اگر اشتباه نکنم 150 گرم آب. و حالا همون یک بسته مثلا پاستا حداقل 30 ثانیه اون پاستا شما اون سسش باید بلند بشه تا به اون برسه و همین. حالا اگر که سوالی دارید میتونید خودتون بپرسید یا میتونید از من بخواید که براشون ترجمه کنم بپرسم اگر که دوست داشته باشید. Any questions? Sure. So I think we do not have any questions in the audience. I just want to say that basically my takeaway was us as physics enthusiasts are always trying to, and we always tend to think about all those complicated problems that we don't get to be hands-on, you know, like quantum mechanics or whatever, general relativity, quantum gravity, stuff like that, beyond the standard model. We always are thinking about all these super complicated things. Meanwhile, there are so many beautiful, nice questions like this, which we can answer scientifically using physics and the laws of physics as you did. And I think this is a very grand research that you have done, and I have a lot of respect for it. And
Thank you.
It was really, really nice indeed. And we all really enjoyed your presentation. So once again, if you may, please. Yes, so if there are no further questions, I guess all the games between So yes, we are going to end the session, and thank you once again.
Yeah,
and,
1 second. I stop the sharing so I can say goodbye like
Yes,
normally. It was a pleasure, guys. Have a nice day.
of course.
Bye. Ciao.
Ciao, Ivan. Goodbye.
Ciao.